So hi everyone, and welcome today for our last webinar about our capabilities. I'm Helene Gautier. I'm a neuroscientist and product owner, organ-on-chip at Netri. And today I will present you this capability number six, about compartmentalization and its application to neurological troubles. So who are we? Netri is a French company based in Lyon, and we believe in challenging the way we discover new treatment. We design, manufacture, and sell standardized neuro organ-on-chip. We intend to use neural activity at the network level to translate mode of action of any drugs. So today the market in preclinical research is already looking to speed up those phases, to diminish the clinical failure rate, and to minimize annual testing. So our view is that preclinical research will be about larger, faster, and more predictive data sets generated animal-free. Therefore, the future of organ-on-chip is about increasing the relevance and complexity of our models while maintaining the high throughput capabilities. We would like to achieve this by democratizing organ-on-chips, leveraging existing equipment, and standardizing data sets while preserving and improving relevance. We're doing this by having a team of engineers, of biologists combined with a digital team, and always keeping in mind the user experience. So today, the presentation will be at the interface between our capability of cultures and the application for neurological troubles. So brain architecture is made of several distinct regions, communicating with each other in a very controlled manner. So far, standard methods in Petri dishes do not allow this compartmentalization, and we cannot reproduce network activity and those connections. The solution is to use compartmentalized culture to recreate the anatomic reality and allowing to have different cells, populations that will represent the different brain regions. It's also important to be compartmentalized if we think about neurological disorders. They're complex and often they involve several brain regions, and their physiopathology is still not fully understood. As an example, in early Alzheimer's disease will start in a specific brain region where neurons communicate with each other and will spread through the brain involving different brain areas. By using our compartmentalized device, we enable to apply injury to only one cell population or one cell area and study the effect on both populations or on the whole cell. Compartmentalizing also allows to study disease progression from one compartment to the other. So, how to record injury consequence on neural network? Well, the solution is to use one of our compartmentalized device with synaptic compartment to recreate a network and to put these devices on top of an MEA plate for functional and also supernatant analysis. So you will have a first compartment with the neural cell body of your first population that will connect in the middle via synapses to the third compartment when you have your neural cell bodies. Our technology with microchannel enhances the 3D isolation and ensures there is no spillover between compartments. And thanks to our partnership with Action Biosystem, this is compatible with MEA and electrophysiology. This is how it looks like our NeoBento developed with Action Biosystem that is so compatible with MEA. And so far, we have 3 of our neurofluidic devices that are coupled on those plates. So the application. We show in partnership with ETAP-Lab that our compartmentalized co-cultures system can be used to understand the cause of Aβ42 aggregation and also its mode of action. So the first step was to use our compartmentalized device and test the action of those oligomers made by ETAP-Lab. At the bottom left, you can see the dose response of cytotoxic assay and in a range from 1 to 10 micromolar of the compound. 1 micromolar, there is no effect on cell viability. However, when tested on functional activity, and this is the right pattern on the screen, you can see on the top in the control conditions that you have nice synchronized network activity. That is the vertical lines that you're seeing on the plot. And on the bottom plot is after treatment, there is a complete disruption of the synchronized activity. So with our devices, we can record an activity disruption that are prior to the cell death. And this could be very important in terms of Alzheimer's disease when it's already too late, when people are diagnosed, neurons are already dead. So all this work is done with ETAP-Lab. And I'm very pleased today that we can have the testimony of Nicolas Viol, CEO of ETAP-Lab, which is a preclinical CRO, renowned for his expertise in the field of dermatology and neurology.
Hi, I'm Nicolas Viol, the CEO of ETAP-Lab, a CRO providing preclinical pharmacology services to pharmaceutical and biotech companies in the field of neuroscience and dermatology. For a few years, our goal has been to complete our in vivo portfolio with new relevant in vitro models of neurodegenerative diseases. To be the most translational as possible, our models under development are based on the use of human iPS cell culture. They are derived into neurons or astrocytes and exposed to toxic soluble oligomers such as tau protein oligomers. It's a key component of Alzheimer's disease. We also want to model synapses and network connections corresponding to brain structural architecture and functions. The organ-on-chip technology offered by Netri allows to achieve the fluidic compartmentalization that is mandatory to create these models. First, it allows to compare the effect of toxic oligomers on treatments on the several parts of the neurons, namely cell body and neurites. It allows to evaluate oligomer spreading by following their transfer from synaptic compartment to body cell compartment. And third, it allows to record the functional consequences of oligomers on treatments on the network by using electrophysiology. As a solution, we use Netri's dual-link MEA and NeoBento to define network alteration and to target or stimulate specific cell parts or connections. With these models, we will be able soon to better evaluate the efficacy and the underlying mechanism of drugs in development in the field of neurodegenerative diseases. Thank you.
So thank you, Nicolas, for the testimony. And to summarize, our technology allowed to create a compartmentalized microenvironment to ensure that we can apply different culture media, ensure appropriate differentiation and maintenance of cells, and co-culture of neurons or non-neuronal cells. We have improved 3D isolation to ensure there is no leakage, to ensure reproducibility as well in our readouts, and also intra-individual reproducibility. Last, the functional compartmentalization, which is an important feature, allows to reproduce functional connection, to apply localized injury to specific cell type or cell area, the cell body versus the neurite, to couple the functional analysis with phenotypical analysis by doing immunostaining post-recording, and we can investigate the injury's impact on functional activity, synapse formation, and network dynamics, while also monitoring the disease progression from one compartment to the other. So thank you, everyone. This is the end of our first series of webinars around our capabilities. The replays of the 6 webinars are available on our YouTube page and on our website, resource page video section. And now it's time for you to tell us what you think and to say if you like the format, the concept, and please don't hesitate to send us feedback and to test you for our next series of webinar where it will be up to you to choose a topic. Last, if you want to benefit from our end of the year discounts, please contact our team at sales@netreport.com. And now, thank you again. And if there is any question, I'm here to answer.